Hi everyone, welcome to the second lecture of Chapter 6, the skeletal system. In the previous lecture, we looked at cartilage and we looked at gross anatomy of bone. Today we will begin looking at the microanatomy of bone, or the histology of bone, and a little bit of how bone growth works, and then we will kind of build on that in our third and final lecture of this chapter. So, as always, let's go ahead and start with our not really attendance questions. Why might cartilage heal slower than bone when there is damage to both? And explain how the level of melanin has to maintain a balance between blocking DNA damage and allowing vitamin D production. So go ahead and pause the video, try to answer those. This might take a little bit more thinking than some of our more traditional questions, especially the second question here. So it's you can kind of think of it as a case study almost, or a thinking cap question. Okay, so we've gone ahead and paused the video, hopefully. Let's take a look to see what the answer to these might be. Why might cartilage heal slower than bone when there's damage to both? Now, this isn't always the case, but usually it is. If you were to damage knee cartilage or damage bone in the knee, chances are the bone is going to heal faster than the cartilage. And the reason is because, remember, cartilage is avascular, while bone is vascularized. Bone has a blood supply, cartilage does not. And blood carrying oxygen and nutrients, things like that, will stimulate the bone to heal quicker. The cartilage can take a really long time to heal depending on the extent of the injury. Sometimes cartilage doesn't heal if it's severe enough. Next, explain how the level of melanin has to maintain a balance between blocking DNA damage and allowing vitamin D production. This is a little bit of a thinker, so let's see exactly what's going on here. Remember, melanin blocks ultraviolet radiation after damage to the keratinocyte DNA has occurred. Those keratinocytes release stimulation hormones that trigger melanin to be produced. So melanin is blocking ultraviolet radiation to prevent further DNA damage. But remember, we said that cholesterol in the skin requires ultraviolet radiation to hit it, and then it is converted to vitamin D, or a precursor to vitamin D. Well, that means that we can't completely block ultraviolet radiation, otherwise we would become vitamin D deficient. So the level of melanin, really we could think of this as the skin color, is a balance between blocking ultraviolet radiation and production of vitamin D. That is why the closer to the equator you get, the darker the skin tone. The further from the equator you get, the lighter the skin tone. At the equator, there is very, very strong ultraviolet radiation, so we need a lot of melanin there. There's still enough ultraviolet radiation getting through to activate vitamin D because there's so much ultraviolet radiation there. But if we go up, say, towards Scandinavia, everybody has really, really pale skin. That's because there's not a lot of ultraviolet radiation up in those areas of the globe. So we want to maximize the amount of ultraviolet radiation that does get through. We don't need a lot of melanin. We don't want a lot of melanin. So there's still enough ultraviolet radiation getting through to activate vitamin D, but we don't have to worry quite as much about DNA damage. So skin tone really is a balance of block enough ultraviolet radiation so that we don't all get skin cancer, but allow enough ultraviolet radiation so that we don't all become vitamin D deficient. Think about that. That's kind of a tricky concept, so listen to that a few times just to make sure that it's sinking in. And with that, let's go ahead and dive into microscopic anatomy of bone, and we will begin with the different types of bone cells. Here on the left, we see something called an osteogenic cell. Let's break that word down. Osteo, remember, is bone. Genic is birth of or the beginning of. So osteogenic cells are stem cells. And they are stem cells that produce other bone cell types. When an osteogenic stem cell divides, one cell remains a stem cell, and the other cell is going to become some other type of bone cell. What type? Well, when an osteogenic stem cell divides, we said one remains a stem cell, the other will either be something called an osteoblast or an osteoclast. First, let's look at an osteoblast. Remember what that root word means, blast? To give birth to build. So osteoblasts are the cells that build bone. They secrete stuff that becomes what we think of as bone. We say they are matrix synthesizing cells. So they are converting non-bony matrix to bony matrix. We'll look at that more in depth in a little bit. But these cells are creating bone. These cells over here, osteoclasts, are exactly the opposite. Clast means to break down. I think of clast as crush, C, clast, crush, blast, build. So osteoclasts are bone resorbing cells, and resorb or resorption is what we call it when we take bone and we break it down. Now that sounds like it might be a bad thing, but it's not. We are constantly building bone and resorbing bone. More about that in our next lecture, but know that both of these things are good as long as they are roughly in equal levels. So osteoclasts break bone down, osteoblasts build bone. Both of them come from osteogenic stem cells. Now, as osteoblasts are building bone, they are laying down what becomes bone. Sometimes they will become trapped. They will become surrounded by bony tissue. And when that happens, they go through a change. They no longer lay down more bone. They stop. And they go through a change and become something called an osteocyte. Osteocyte. Osteo, bone, cyte, cell. These are just regular bone cells. They are not building bone. They are not breaking bone down. They are living inside of bone. If you've already looked at the osteon in lab, these are the cells that you saw there in the lacuna, which we'll talk about in just a little bit. So osteocytes are mature bone cells. They maintain that already formed bone. So osteogenic stem cells divide. One cell stays an osteogenic stem cell. The other cell becomes either an osteoblast or an osteoclast. If it becomes an osteoblast, it's building bone, building bone, building bone until it becomes trapped, at which point it converts and becomes an osteocyte. Those are the different types of bone cells that we find. So now let's zoom out a little bit. And this picture can be tricky because it's round and it has a hollow center. And that confuses students what it is that they're seeing. This part that's pulled forward right here is called an osteon. You probably have already seen that in lab. Here is another image of it seen from a different angle. Here is what we saw in lab down in the bottom right. Now, what we are seeing here is microscopic. This is not the shaft of a bone that's sticking forward right here. This is much smaller than that. Look up here at the top of this image on the right. This is a bone. This small little section at the border of the diaphysis is what we see in this large image right here. So this part right here where it says spongy bone, this is the wall of the medullary cavity. So this is the hollow part of that bone. Now we are looking here at the edge of the diaphysis. So this little part that's pulled forward right here is much too small to be seen with the naked eye. Okay, so hopefully we've kind of got that now in our head. We can tell what it is that we're looking at. Let's talk about the features, the structures that we see at this very microscopic level. Like we said, pulled forward here is an osteon. And if you've already had lab, you've seen the osteon model. But we can see that there are layers to this osteon. And these layers are called lamella. Lamella. Lamella are these layers that we see in bone. They kind of look like tree rings. Specifically, these lamella are called concentric lamella. Concentric lamella. Now let's take this entire osteon out. Now this doesn't happen, we're just kind of using our imagination here. Let's take this osteon out of that bone, and that's what we see over here on the left. It looks a little bit different because we see these blue stripes, which are not actually there. They're just here to show us the orientation of something in these lamella. Collagen fibers. The same collagen that is in our skin, the same collagen that is in cartilage, it is also in bone. And the orientation of the fibers, we can see they're going kind of diagonally up towards the left in this layer, but in the next layer they're going diagonally up towards the right. And then in the next layer they're going diagonally up towards the left. So the collagen fibers run in different directions. They're in alternating directions at each layer of that lamella. That is what gives our bone such strength. That's why when we're walking, our legs don't buckle beneath us. The orientation of the collagen fibers help resist shock and tension and twisting forces and things like that. Now, if we look in the middle of the osteon, there's this opening called either the central canal or the Haversian canal. Again, this is not the medullary cavity of a bone. This is much smaller than that. These are these little openings right here. Inside the middle of each osteon is an opening called the central canal or the Haversian canal. And inside that, we have our blood vessels. Very small arteries, very small veins. We don't see it in the image here, but there are usually lymph vessels. And there is a nerve fiber. Those run the length of the bone through the Haversian canal. Here we've cut one of them open so that you could see how they are oriented. They run the length of bone. Now, very, very similar to that is something called either a perforating canal or a Volkmann's canal. It is still an opening where we find blood vessels, lymph vessels, and nerves, but they run perpendicular to the bone, perpendicular to the osteons. They connect neighboring haversian canals. So here we can see blood vessels running this way down this haversian canal, and then they can take a turn and go into this other haversian canal. Well, how do those blood vessels get in there? Remember from the previous lecture, we have that periosteum that has blood vessels in it. And then we have those nutrient foramen. Well, that's where it perforates through the periosteum into the bone itself, and then into these perforating canals and Volkmann's, perforating canals and central canals. Now here, down here in the bottom left of the right image, we're zoomed in even more. This is one layer of a lamella, and another layer of a lamella, and another layer of a lamella. Here we can see within some bone cells. These are those trapped cells, the osteocytes. And the osteocytes live inside of a little pocket called a lacuna. We saw these in the osteon model. The osteocytes live inside of a pit called a lacuna. Here's an artist's drawing. Over here we can see, this looks like an electron microscope image, and over here is a light microscope image. So the osteocytes are trapped inside of lacuna. They are pretty much completely surrounded by bony tissue. But they are not completely isolated. We need to get nutrients and oxygen from the blood supply into those cells. And we need to get waste and carbon dioxide from those cells into the blood supply. So there has to be an even smaller path from those blood vessels to each of these osteocytes. And there is. These osteocytes have small little projections that reach toward the blood supply and outward towards their neighbors, where they connect with their neighbors. These little projections are extensions of the cytoplasm, and they connect to neighboring cells. These are called canaliculi. Canaliculi. That means little canal. Here in this image, we can see that each cell is connected to neighboring cells using those canaliculi. Over here in the right, this is what we saw in lab when we looked at the bone slide. These little very, very small black lines, those are canaliculi. Now the canaliculi are extensions of the cytoplasm from one cell, extensions of the cytoplasm from another cell that meet. And where they meet, we find, see if you can guess, gap junctions. Think back to chapter 3. Gap junctions are a type of cell junction that's kind of like a tunnel that allows substances to flow between two cells. So we have nutrients and oxygen moving out of an artery into a cell. It travels through those canaliculi, through those gap junctions to the neighboring cells. And waste and carbon dioxide does just the opposite, moving towards this vein here. All right, so we can see all that we've just talked about in this image here, and we see these layers of the lamella that go around a haversian canal. But an osteon is round, and if you stick a bunch of circles next to each other, it doesn't form a nice, complete border. There's going to be spaces in between. Well, those spaces in between are not empty. We still have lamella there, but each lamella that we find there does not belong to a specific osteon. For example, this one that's indicated by these brackets here. All of these rings around it belong to that osteon. All of these rings here belong to this osteon, and all of these rings here belong to this osteon. But there's lamella here that do not belong to any of those osteons. These lamella fill in the spaces between neighboring osteons. These lamella are called interstitial lamella. Interstitial lamella fill in the spaces between neighboring osteons. And finally, running around the entire bone is another type of lamella called a circumferential lamella. Circumferential lamella surround entire bone. All right, so now instead of getting bigger, let's get even smaller, smaller than bone cells. So let's look at the chemical composition of bone. If we were to look with a strong enough microscope, we would see not only are there bone cells, but there's a lot of bony matrix. So let's look at what makes up that bony matrix. When we look at bone, there are two different major parts. There's the organic components, remember, things that contain carbon, and the inorganic components, things that don't contain carbon. Well, the organic components, well, obviously they're cells, but we're looking smaller than that. The inorganic, or the organic components of that bony matrix we call osteoid, and the inorganic components we call hydroxyapatites. So let's look at the osteoid. Osteoid is the kind of gelatinous extracellular matrix that contains things like proteins. The inorganic components, the hydroxyapatites, are mineral salts. These are things like the calcium and the magnesium and the phosphate, things like that. This is what gives bone that kind of crystalline feel. If we did not have the hydroxyapatites, if we didn't have those mineral salts, the organic components, if we only had osteoid, bone would be very gelatinous, very jelly-like. So now let's talk about how bone is formed. Where does bone come from? It's called ossification. Ossification is the formation of bone. And all bone begins as cartilage. Different types of bone begin as different types of cartilage, but all bone begins as cartilage. First, what we see here is called endochondral ossification. We'll look at the other type in just a moment, but this is endochondral ossification. Now, there's a lot of information on here, and I have room for all of these notes on your outline. You do not need to know all of these steps. I have them there because it's interesting, but you do not need to know it. So we are not going to go through in very much detail at what happens at each step. We're just going to kind of look at the big picture. Endochondral ossification begins with a hyaline cartilage model. We have basically a bone. Really, we have basically a skeleton made out of hyaline cartilage. And now this is early on. This is in the womb. And this is what happens to all bones that are below the skull except for the clavicles, the collarbones. All of the other bones below the skull except for the collarbones use endochondral ossification, beginning with a hyaline cartilage model. And I'm going to really quickly run through what happens here. And again, you do not need to know these details. What I want you to know is endochondral ossification uses hyaline cartilage, and it makes all of the bones below the skull except for the clavicles. That's what I want you to know. The details, not so much. So here we see that hyaline cartilage bone. Kind of a funny way of saying it. It's not a bone yet. It is a bone model made of hyaline cartilage. And we get something called a primary ossification center. This is where the seed begins to grow. This is where the bone formation starts. We start converting some of that hyaline cartilage into bone. How do we do that? Well, that's what we were kind of hinting at right here. We have these cells that begin to lay down osteoid and hydroxyapatites. That cartilage begins to become a little bit bony. As that happens, the bone begins to hollow out. That's when we start to get the actual diaphysis and medullary cavity of the bone. And around this time, blood vessels start to penetrate. And once blood vessels show up, now everything kicks into high gear because we have those nutrients. Everything can move a lot quicker. We start to get some spongy bone here lining the medullary cavity. Once we get enough of that forming, more blood vessels grow. We start to have the same thing happening at each end of the bone at the epiphysis. And eventually, all of that cartilage is converted to bone except for at the epiphyseal plate, the growth plate, and that articular cartilage. In those two places, we still have hyaline cartilage. Now, this is for a long bone, but in short bones, it's a little bit different. Irregular bones, it's a little bit different, but it still follows the same basic path. And again, what you need to know is endochondral ossification begins with hyaline cartilage models, and it happens in all of the bones below the skull except for the clavicles. The other option is something called intramembranous ossification. This happens in flat bones, the bones of the skull and the collarbones, the clavicles. And it uses a fibrocartilage model. And that's what I want you to know about intramembranous ossification. Uses fibrocartilage and happens in flat bones, the bones of the skull and the clavicles. Pretty much the same thing as we just saw before. I'm not going to go through all the details. There are some differences, but you do not need to know all of these details for our class. Just a reminder, the joint chapter is self-study. You need to know how joints are classified from 8.1 to 8.3. You need to know the structure of a synovial joint. You need to know movements. You need to know the types of synovial joints from 8.1, and you need to know the disorders from section 8.6. Well, what about after we're born? We don't stay, the size of an infant. We continue to grow. So postnatal growth of bone happens also. How do we get taller? Well, growth in length happens at the epiphyseal plate, the growth plate. There's one at each end of a long bone. Here we can see we are zoomed in at the epiphyseal plate. This is one of the epiphysis of the long bone. Down here is the diaphysis and the medullary cavity. So we are looking right here near where the epiphysis and the diaphysis meet. This part, throughout childhood, throughout the early part of your adolescence, is still cartilage. It's hyaline cartilage, like we just saw. And now we're going to take this area and zoom in, and that's what we see right here. This is a microscopic image of the epiphyseal plate. This is where growth in length of our long bones occurs. So there are different regions. Even just looking at this, if there weren't everything written over here on the right, you could see that the top, here in the middle, down near the bottom, it looks different. And that's because there are different things there, different things happening. So up here, nearest the epiphysis side of the bone, we have something called the proliferation zone. This is hyaline cartilage. We can see the cells are very small. They are undergoing almost constant mitosis. And as they make more and more cartilage cells, those cells are pushed towards the diaphysis, towards the shaft of the long bone. until eventually we get to the next area called the hypertrophic zone. And we can see here the cells in their lacunae are much bigger than they were up here at the proliferation zone. The proliferation zone is still undergoing mitosis, but those cells that are being produced are pushed down into the hypertrophic zone where the mitosis stops, but they get bigger. As they enlarge, well, proliferation zone still up here going through mitosis, pushing more cells into the hypertrophic zone, which pushes those enlarged cells into an area called the calcification zone. And here, that bony matrix starts to be laid down. We start to lay down those hydroxyapatites. So the matrix, we say it calcifies. And as that happens, those chondrocytes, the cartilage cells, begin to die. The matrix begins to deteriorate. It starts to break down. We start to see cavities. And as that happens, blood vessels now have room to grow. So blood vessels move into those cavities, and that then triggers bone cells. Osteogenic cells, osteoblasts, osteoclasts, they all become active. And here in the ossification zone, new bone is being formed. Meanwhile, all of that we just went through is still happening. Proliferation zone pushes down into the hypertrophic zone, pushes down into the calcification zone, pushes down into the ossification zone. That goes throughout, childhood, early adolescence, at which point it begins to slow. But growth in width will continue throughout lifetimes. Bones will continue to get wider. And what happens is stress on bone. Now, that's not a bad thing. Stress on bone is what causes bones to get wider, to get denser, to get thicker. The more stress is put on a bone, the stronger it will become. So someone who, let's say, is bedridden, this is something that always bothers me when you watch TV or movie and someone's been in a coma for, 3 years, and then they wake up and they get up out of bed and they stand up. Well, that would not happen. One, their muscles would be atrophied, so they wouldn't be strong enough to support them. And two, there's been no stress on their bones, so their bones would shatter beneath them. But if you look at, say, a weightlifter who's constantly putting stress on their bones, if you were to look at their bones, they would be very, very dense, very, very thick. So stress causes growth in width. Okay, so hormonal control of bone growth. Bone growth is under the control of a few different hormones. First, let's talk about growth hormone, which is produced by a gland in your brain called the anterior pituitary gland. The anterior pituitary gland makes a bunch of different hormones. One of them is called growth hormone, or GH. And growth hormone is what triggers bone growth, sort of. We'll get to that in just a minute. But the amount of growth hormone is partially regulated by the thyroid gland. There's something called thyroid hormone, or T3, T4, or thyroxine. Thyroid hormone regulates the levels of growth hormone. But growth hormone, even though we think of it as causing skeletal growth, it doesn't really stimulate bone growth. What it stimulates is cartilage growth and the conversion of cartilage into bone. So growth hormone does not stimulate bone growth, it just triggers the growth of cartilage and the conversion of cartilage to bone. Also, the sex hormones, testosterone and estrogen. Testosterone has a much bigger effect on the growth of bone, but estrogen also does to a lesser degree, and estrogen also triggers how we handle calcium. So when we hit puberty, we have a growth spurt. And that is because when we hit puberty, there is a sudden spike in testosterone and estrogen. And by the way, both males and females have testosterone and estrogen. A lot of people think males testosterone, females estrogen, but no, both males and females have both estrogen and testosterone. So as estrogen and testosterone spike during puberty, we have a sudden dramatic increase in this epiphyseal plate activity. That's why we have that burst that we call a growth spurt. And then into adolescence, we start to see those estrogen and testosterone levels begin to taper off, begin to come back down. They don't go away. They just go from that very heightened level that we had at puberty back down to a more normal lifelong level, which then causes what we say the growth plate will seal. What that means is those chondroblasts slow at adolescence. Up here in the proliferation zone, they begin to stop dividing, which stops pushing into the hypertrophic zone, which stops pushing into the calcification zone. The ossification zone takes over, and the growth plate seals at adolescence. All right, so here, let's take a look at what happens when growth hormone is in excess. Over on the left, we see two people. The man on the farthest left is an average height male. On the right, this is a man with something called gigantism. Gigantism is what happens when you have too much growth hormone from childhood. You get really, really tall. Over here on the right, well, we have two guys. Over here on the left of the right picture, we have a man who is of average height. And on the right, we have another man who is of average height. If we look at the man on the right, though, we could tell something is going a little bit different. This man has something called acromegaly. Acromegaly. This is a condition that comes about when you have too much growth hormone, but it's not from childhood. This happens later in life, sometimes after childhood, after he stopped growing. Something, either a blow to the head or a brain tumor, triggered growth hormone levels to be too high. And if he was not wearing this robe, we would see, well, one, his head is very enlarged. His nose is very enlarged. His jawline is very enlarged. We can't see his joints, but his elbows, his wrists would be very enlarged. We can see his hand is very enlarged. His thumb is very big in diameter. Same thing would be going on his legs. His hips, his knees, his ankles would be very enlarged. That is acromegaly. But he's the same height as this other man here. Over on the left, gigantism. Acromegaly on the right. Gigantism is too much growth hormone from childhood. Acromegaly is too much growth hormone after adolescence. So let's use this as a thinking cap question. Oversecretion of growth hormone from the anterior pituitary, we say hypersecretion of growth hormone, results in gigantism or acromegaly depending on the age of onset. Why are the two conditions different? Why is this man on the right not really, really tall like the man over here with gigantism? What's the difference? So think about that. Don't just wait until I answer it. See if you can come up with the answer here. Well, in gigantism, It was from childhood. He grew really, really tall. Why? Because his growth plates were still open. If your growth plates are still open, when there is increased growth hormone, they're going to become very active, and he's going to continue to get taller and taller and taller, reaching a very excessive height. Over here on the right, his growth plates had already closed before growth hormone hypersecretion. It doesn't matter how much growth hormone there is. He cannot get any taller because his growth plates have closed. But we can still get that growth in width. And that's what we see here. It causes the bones of the head to get much thicker. It causes the bones to get thicker, which is really noticeable around the joints. So acromegaly is here. All right, so that's the end of today's lecture. We will pick back up with the third and final lecture of Chapter 6 next time. If you have any questions as you listen, please reach out. Please let me know. Otherwise, I will talk to you in our next lecture. Take care.